In the previous video, we went over the general pattern of how viruses enter a cell and use that cell to replicate themselves. But some viruses, the retroviruses, do this process a little differently. One retrovirus that you're probably familiar with is HIV, the human immunodeficiency virus. The HIV virus attacks white blood cells, weakening the immune system. If not treated, this can lead to acquired immunodeficiency syndrome, or AIDS. Let's make sure you caught what the HIV virus actually infects. So what makes a virus a retrovirus? First of all, retroviruses are RNA viruses, which means they mutate quickly. They also contain an enzyme called reverse transcriptase. When we go through the steps of viral replication in a retrovirus, you'll see why reverse transcriptase is important. It adds some extra steps in which instead of just replicating its RNA, the virus uses reverse transcriptase to build viral DNA from the viral RNA. Just to refresh, here's the basic viral life cycle we talked about before. A virus that is not a retrovirus will attach to the host cell and enter it. Then after the capsid comes apart, it uses the host cell's ribosomes to make copies of its own genetic material. Then the new virions assemble and are released from the host cell. With retroviruses, the main difference will be in this third step, the replication process. We're going to go through this process and the extra steps in replication of a retrovirus. But here is a list of steps in each process, just to show you the differences and help you remember the order of operations. Okay, so the process starts the same as any other virus. First, some glycoproteins on the outside of the retrovirus bind to receptors on the host cell. The virion then enters the host cell, injecting its RNA and some proteins like enzymes. One of those enzymes is the reverse transcriptase that we mentioned before. Here's where the process gets different. Instead of just replicating the viral RNA, the enzyme reverse transcriptase is used to make viral DNA from the viral RNA. So the name helps you remember what reverse transcriptase is doing here. It's reversing the transcript, right? Again, converting that RNA into viral DNA. So now we have viral DNA in the host cell. Now another enzyme is going to come into play. The enzyme integrase is used to integrate that viral DNA into the DNA of the host cell. So again, it's doing exactly what its name implies. Now we have this hybrid DNA molecule with the host cell's original genome and the viral DNA added in. So what's happened here is the retrovirus has essentially genetically modified the host cell to produce new viral RNA and viral proteins. When the host cell uses its DNA to produce RNA, it is now also making copies of the viral RNA, which can be used to make viral proteins. And then, just like in the general virus life cycle, those new proteins and viral RNA assemble together near the edge of the host cell. And then new virions are released by budding, so they can go out and infect new cells. It can be difficult to see the differences in typical virus replication and retrovirus replication. You may want to draw out these processes side by side to understand what's going on in each. When you're done with that, try answering this question. This process is complicated and has a lot of steps to understand. So your activity for the content on Exam 1 will focus on the replication of retroviruses. Under the Week 2 module, you'll find an assignment called Activity 1. This is a PowerPoint file with objects that you can move and drag to the correct locations to fill out the diagram. Use your notes and think through this process to correctly fill out the diagram. You will use all the objects in the box in the PowerPoint file. When you're done, save the file as a PDF and upload that to the eCampus assignment.